This is Jessie Hunt, Chapter 9 Outline, Segment 1, pages 263 to 271. This chapter considers sensory systems which allow us to monitor important signals from a distance. These systems include the auditory system, hearing, the olfactory system, smelling, the vestibular system, movement of the body, and the gustatory system, tasting. The basics of sound, box 9.1. The sound is the brain's interpretation of patterns of local increases and decreases in air pressure, usually caused by the vibration of objects. A cycle is a single alteration of compression and expansion of air. Pure tones are a tone with a single frequency of vibration. A pure tone is described in two ways. Its amplitude or intensity, which is measured as air pressure, or the force per unit area, dynes per centimeter square, is the common term is loudness. And frequency, the number of cycles per second, measured in hertz. The common term for this is pitch. Most terms contain more than one frequency. A fundamental frequency is the predominant frequency of an auditory tone when it has multiple frequencies in it. Harmonics are multiples of a fundamental frequency. Timbre is the characteristic sound quality of a musical instrument due to its relative intensity of harmonics. The Fourier analysis is a mathematical process of taking a complex wave and decomposing it into multiple simple sine waves. Decibels are the logarithmic scale used as a measure of sound intensity. Hearing. Sound and hearing have been an integral part in evolution and the survival of organisms. Pressure waves in the air are perceived as sound. Evolution has shaped our auditory system to hear sounds of biological importance to us. The sound is guided into the inner parts of the ear. The mechanical energy of the sound has to be transduced into neural activity. Transduced is the conversion of one form of energy to another. The external ear captures, focuses, and filters sound. The external ear consists of two parts: the pinnae, the external readily visible part of the ear, which funnels sound waves into the ear canal, and the ear canal, which is a tube leading from the pinna to the middle ear. Many animals can swivel their ears. The pinna modify the sound before it travels to the middle ear. Humans' ears enhance the sounds between 2,000 and 5,000 hertz. These are sounds which are important for speech perception. The shape of the external ear is also important for detecting direction and distance of sound. The middle ear concentrates sound energies. The middle ear is the cavity between the tympanic membrane and the cochlea. The tympanic membrane, or the eardrum, seals the end of the auditory canal and is the partition between the external ear and the middle ear. The ossicles. These are three bones which transmit sound across the middle ear from the tympanic membrane to the oval window. The malleus is the first one. It's the hammer, and it's connected to the tympanic membrane. The incus is the anvil, which is between the malleus and the stapes. And the stapes is the final one. It's called the stirrup, and it's connected to the oval window. Two muscle groups improve auditory perception and protect the delicate inner ear from the potentially damaging sounds of the external ear. These two muscles are the tensor tympani, which attaches to the malleus and modulates the mechanical linkage between the malleus and the tympanic membrane to protect from damaging the receptor cells of the inner ear, and the stapedius, which is attached to the stapes. These muscles will stiffen when a noise is heard and will reduce the amount of physical movement inside the ear. This protects the ears from being damaged by loud noises. This is a reflexive process. Just before we create an internal noise, the tensor tympani and stapedius stiffen. This is why internal noises are not deafeningly loud. The cochlea converts vibrational energy into waves of fluid. The inner ear is the cochlea and vestibular apparatus. The cochlea is a snail-shaped structure in the inner ear which contains the primary receptor cells for hearing. It is filled with fluid. It measures about 4 millimeters in an adult human. It consists of 3 parallel canals which run the length of the cochlea. These canals are the scala vestibuli, or the vestibular canal, the scala media, or the middle canal, and the scala tympani, or the tympanic canal. The round window is a membrane which separates the scala tympani from the middle ear cavity. Organs of Corti is a structure in the inner ear that lies on the basilar membrane of the cochlear. It contains hair cells and terminations of the auditory nerve. It consists of three main structures. One, sensory hair cells, two kinds. The inner hair cells, which are in a single row closer to the central axis of the cochlea, and the outer hair cells, which are in three rows, and each hair has 50 to 200 stereocilia. It also consists of a framework of supporting cells and the terminations of auditory fibers. The basilar membrane separates the scala tympani from the scala media and vibrates in response to sound. The stapes movement causes the liquid in the scala vestibuli to create waves which ripple the membrane, the basilar membrane. The rippling of the basilar membrane, of the hairs on the basilar membrane, which ripples the basilar membrane and ripples the hairs on the basilar membrane. The basilar membrane is tapered. It's wider at the apex of the cochlea and narrower at the base of the cochlea. Low-frequency sounds ripple more at the wider, more flexible parts of the basilar membrane. High-frequency sounds ripple the membrane at the narrower, stiffer parts of the basilar membrane. The heights of the stereocilia increase progressively across the hair cell so that the top forms a slope. Stereocilia are a relatively stiff hair which protrudes from the hair cell in the auditory or vestibular system. Tectorial membrane sits on top of the organ of Corti in the cochlear duct. Auditory nerve fibers connect the hair cells at the base. The organ of Corti has four kinds of synapses and nerve fibers. They have afferents, which convey messages from the hair cells to the brain, 1 and 2, and efferents, which convey messages from the brain to the hair cells, 3 and 4. Each IHC is associated with about 16 to 20 auditory nerve fibers, and these account for 90 to 95% of all afferent fibers. OHCs have a relatively small amount of auditory fibers connected to them. OHCs do not detect sound. They push on the tectorial membrane in response to commands from the brain via efferent nerve fibers. They do this to fine-tune the organ of Corti. IHCs also receive efferent signals, most likely for inhibitory purposes. How do IHCs transduce sound into neural activity? As the basilar membrane ripples, the vibrations cause the stereocilia on the hair cells to bend. Small displacement of hair bundles causes rapid changes in the ionic channels of the stereocilia. Each hair cell has only about 100 ion channels and about 1 to 2 per stereocilia. The channels are located at the top of the stereocilia. When the stereocilia sway, the tension in the tip links increase. The tip links are a fine thread-like fiber that runs along and connects the tips of the stereocilia. When tension is put on the tip links, they pop open ion channels to which they are attached. The channels snap back closed once the stereocilia sway back into place. Opening the channels allows potassium and calcium to rush into the cell. This causes rapid depolarization of the whole cell. The initial depolarization causes an influx of calcium at the base of the hair cell. This influx of calcium at the base of the hair cell causes vesicles to fuse with the presynaptic membrane and release neurotransmitters, probably glutamate. The action potentials then travel to the brain via the vestibulocochlear cranial nerve. Active mechanical processes in the cochlea enhance frequency discrimination. Humans can discriminate between sounds with a difference of about 2 Hz. OHCs help fine-tune the cochlea and discriminate frequencies. Hyperpolarizations cause OHCs to lengthen. Prestin is a motor protein which allows OHCs to change length. Depolarization causes OHCs to shorten. This allows for pronounced amplification and sharpened tuning. The cochlea produces sounds of its own because of the mechanical movement of the basilar membrane and by pushing back on the eardrum. Autoacoustic emissions is a sound emitted by the cochlea spontaneously or in response to an environmental noise. Auditory signals run from the cochlea to the cortex. There are about 30,000 to 50,000 auditory fibers on each side of your head. A tuning curve is a graph of the responses of a single auditory nerve fiber or neuron to sounds that vary frequency and intensity. The auditory nerve fibers on either side of your head split into two main branches going to different locations as they enter the brainstem. They go to the dorsal cochlear nucleus and the ventral cochlear nucleus. The auditory fibers split again in the cochlear nuclei. One path from each nucleus goes to the superior olivary nuclei. The bilateral input is where the first binaural effects are processed. This has to do with localizing sound. Several other paths converge on the inferior colliculi, primary auditory centers of the midbrain, which is the primary auditory center of the midbrain. Outputs from the inferior colliculi go to the medial geniculate nuclei of the thalamus. This sends projections to auditory cortical areas.